Thank you, Dr. Matoba. The Center for Integrative and Environmental Health Sciences and the Department of Pharmacology and Toxicology welcome you to our Environmental Health Sciences Series seminar. Today's speaker is Dr. Petra Haberzettl, Assistant Professor of Medicine in the University of Louisville School of Medicine and Associate Director for the Center for Integrative and Environmental Health Sciences. Dr. Haberzettl earned her diploma in biochemistry from Ruhr University Bochum before earning her PhD from the Institute of Environmental Medical Research at the Heinrich Heine University in Dusseldorf, where her doctoral thesis investigated the role of actin cytoskeleton in Fc gamma receptor 2 particles uptake in the alveolar macrophages. Dr. Haberzettl joined the University of Louisville as a postdoctoral fellow in 2007 before being appointed to her current position in 2011. She's a well-respected researcher in the field of inhalation toxicology, having published 47 peer-reviewed journal articles and receiving the 2019 Young Investigator Award from the Society of Toxicology, Inhalation and Respiratory Toxicology Specialty Section. Dr. Haberzettl's research investigates the effects of PM2.5, volatile air pollution, circadian rhythm disruption, and obesity on development of type 2 diabetes and cardiovascular disease, and her research is currently supported by a CIEHS pilot award. Today, Dr. Haberzettl will be presenting a talk titled Chemical and Non-Chemical Environmental Determinants of Cardiovascular and Metabolic Health.
Thank you for this nice introduction by an award-winning student. What an honor. I want to welcome everybody here for my presentation. I switched it a bit up this time, so it's not traditionally my start with like air pollution in the first place. I want to talk first about the non-chemical determinants. And I'm looking for cardiometabolic disease development, and it's really important because we are in the midst of an epidemic of type 2 diabetes, and it seems to rise in the future in almost all of the world. In the US, about 42% are obese and about 12% have diabetes. And it is important to look for these because obesity increases not only the risk for type 2 diabetes, but combined obesity and type 2 diabetes are a major risk factor for cardiovascular disease, increasing the cardiovascular disease risk by 3 times in women and 2 times in men. Traditionally, people thought it's genetics, it's having bad diets, having no physical activity, and these were the major risk factors considered. But more recent research suggest also that environmental factors are significant contributors to the epidemic of type 2 diabetes and cardiovascular disease. So what are these? So we have either chemical or non-chemical contributions, which are contaminants, chemicals in soil, in the water, or in the air. We have also the non-chemical contributions by noise and light pollution, but also due to climate change, increasing temperatures, as well as more stresses due to flooding and other disasters. And not to think that we are off the hook. These are really in the US present everywhere where we go. It is also important to mention that these are mainly overlapped in the stronger urbanized areas in the east and on the west coast. I will focus my presentation mainly on the light pollution aspect and the air pollution aspect, starting with the light pollution. So what we know normally from light pollution is, it's annoying for any astronomer, right? We cannot see skies anymore. So this is like the last one is approximately what I can see from my house. And I'm living in the middle of Louisville, so it's not really nice. I have to go to the Grand Canyon to see something on the left side, right? So it's really annoying for astronomers. I know my husband is one. But there's also other effects which are affecting our health as well. So we have light pollution, which affects the wildlife. We started with sea turtles, which don't find their way to the ocean, but there's also plant life affected. For instance, artificial light at night in the spring leads to an earlier leaf bud break, and in the later fall, the color changes are coming later, which we, I think, experience right now. And this is independent of temperature changes. Of note is also for our personal health that a more active season in urban plants also affects us by having longer pollen seasons and exacerbating the risk for asthma and other breathing problems. Wildlife, so that's the other thing. So I choose the fireflies. These animals really rely on light to find a partner. And when you have artificial light at night, this can lead to desperation, heartbreak, and fatal attacks. So it's important for these animals, but it's not only for the insects important. It's also important for animals who rely on them as food and for plants who need them for pollination. Now back to our approach to look for cardiometabolic health. So this is my window from my bedroom before we convinced our neighbors to change the light bulb. It got a bit better, but we are still exposed to light pollution a lot. And this has been realized to become really harmful for cardiometabolic health and cardiovascular disease. So there's an increase in cardiovascular disease, hospitalization and death, which is similar to this air pollution. It has also been found that artificial light at night increases the resistance, insulin resistance, and decreases beta cell function, all over increasing the risk for developing type 2 diabetes. However, although we know that exposure to either outdoor light or indoor light at night increases the cardiometabolic risk, the mechanisms are still not well understood. So what could be the reason why we have these effects of light? It could be by impacting that artificial light at night impacts the circadian rhythm. The circadian rhythm is the physiological and mental behavior that follows a 24-hour cycle. This is the day-night cycle we are used to for all the years we are on Earth. It's normally controlled by the central clock and regulated by external signals, not only light, but also temperature, food, and physical activity, which are called Zeitgeber. It was initially thought that the central clock is the main regulator, but the discovery of the peripheral clocks leads to a paradigm shift, finding that every little cell in your body has its own clock, a transcriptional feedback loop that regulates circadian timekeeping independent of the central clock. This discovery got awarded with the Nobel Prize in 2017. So how is this normally working? So under healthy conditions, we have the central clocks, which resets all the clocks in the tissues and keep everything aligned and synchronized. When we have, and this is important to regulate daily activities and to get a rest at night, all the physiological aspects are regulated by this interaction between these two. Under conditions where we are exposed to artificial light at night, this doesn't work anymore. So it starts to desynchronize. The central clock is not getting synchronized from the light anymore. And then it cannot synchronize all the tissue clocks, which become more and more misaligned. On the other hand, there's also a disorganizing of the circadian rhythm, which could be occurring when you eat for instance, at the wrong time. So you go in the middle of the night to the fridge, not only that you're exposed to light at night, but eating is a signal which triggers the clocks to reset to a different time point, and they become similar misaligned as in the synchrony case. So both of these, either disorganized circadian rhythm as well as the synchrony, has been associated with cardiometabolic disease. So we had already pointed out that artificial light at night is the main contributor, but there are other lifestyle factors which also disrupt the circadian rhythm, and these are bad sleep habits, shift work, and jet lag. For instance, in human, extended or shortened sleep duration, so sleeping more or less than 9 hours or less than 5 hours per night, shift work as well as forced synchrony, which is a laboratory experiment, have been shown to decrease glucose tolerance and insulin sensitivity and increase the type 2 diabetes risk in human. In mice, changing the light-dark cycle, surgical dealteration of the suprachiastic nucleus, or the genetic disruption of the circadian rhythm have also shown to increase not only inflammation, but obesity, insulin resistance, and also lead to vascular dysfunction. However, the mechanism how these misalignments impair cardiometabolic health are still not 100% clear. That's why we started thinking about that this can be related to inflammation and inflammatory responses, and it has been known that the molecular clocks control inflammatory responses. But in contrast to the inflammatory responses, it is not really understood whether pro-resolving responses, which are clearing up inflammation, are also clock regulated. And for this, we hypothesize that extended light exposure or artificial light at night impairs cardiometabolic health by disruption of pro-resolving pathways, thereby promoting chronic inflammation and insulin resistance. So this is an initial study. So we did the extended light exposure by exposing mice to 6 additional hours of light in contrast to the 12-hour, which is the regular light cycle. Then we measured circadian activity, corticosterone levels in the plasma and clock gene expression that all confirmed that there was a disruption of the circadian rhythm. We also performed then a glucose tolerance and insulin resistance test by either doing a GTT shown on the left side, and the area under the curve shows that in both males and females, glucose tolerance occurs with extended light exposure, and then the HOMA-IR indicates insulin resistance in both sexes. We also looked for expression of enzymes which are triggering the production of specialized resolution mediators, which was decreased by the extended light cycle. And this is only male data. This is our initial study, so we are now progressing and trying to figure out how this happens and try more to tie it to the circadian rhythm. So this was my story about the light pollution, and as you can see here, the development of type 2 diabetes and obesity overlaps with the increasing light pollution we experienced during the years. However, there's another overlap which I'm working on for a really long time. That's between type 2 diabetes and air pollution. Interestingly, this is not related there's no association to obesity per se, but air pollution, PM2.5 exposure has been shown in human to increase the prevalence for type 2 diabetes. So what is air pollution? So we can have either man-made sources for air pollution, which come from industry or just traffic. We have also wood fire burning and farming. But we have also natural sources, and these become more and more important due to the impact of wildfires, as you can see in the lower picture. Even there is no wildfire, it reaches now the big cities. This is New York, but we have experienced this in the last 4 years in Louisville as well, seeing like a yellow sky in the summer because the wildfires from Canada, the air from the wildfires in Canada made it to Louisville. Air pollution consists of gaseous and volatile components, which are important, but I will not focus on those. My main research focuses on the particulate matter fraction of air pollution. And these are characterized by their size. And there we have like ultrafines, fine, and coarse particles, which are different in their way how they can penetrate the body. So the coarse particles can only make it to the upper respiratory tract and get usually eliminated by exhalation, swallowing, or mucociliary clearance, while the fine and ultrafine particles can make it into the respiratory tract and go deep in the alveoli. While it's not thought that the fine particles can make it in the bloodstream, the ultrafine could reach even the bloodstream. Different on these, it's also really important to look for the fine fraction, as these are stuck in the lung and cannot be removed. So these are the ones who are mainly considered to be reasonable for cardiovascular disease. We have an inhalation facility here, which we use to do real-world exposures. These are in the 7th floor of the MDR. So we draw the air from the outside into our inhalation system, where we expose mice in chambers right here. These mice receive either HEPA-filtered air or concentrated particulate matter. And we tested whether the size is appropriate. And we also tested for chemical composition. And we found no difference between the real-world outdoor PM and the concentrated PM, which we have in our chambers. So this guarantees real-world exposure scenarios. Our initial study, which is long time ago, which I did together with Dr. Conklin, was on testing whether PM2.5 induces insulin resistance. By itself, for 30 days, it was not sufficient. It needed another trigger. In this case, we used high-fat diet. And as you can see from the left graph, it increased insulin with glucose intolerance in high-fat diet fat mice, but not in mice fed a regular diet. So from this, we were thinking maybe extending the light cycle does something similar and increases the sensitivity to air pollution. And for this, we had the same protocol for the extended light cycle as before. Only caveat is we now expose the mice to PM 2.5 in the morning for 6 hours. And we did also in another approach, we exposed them a bit later just to see whether there's a difference in the time of the day. We again confirmed that there was a circadian disruption, and here are the filters. So the left one is the HEPA filter air, the right one is the PM 2.5 filter indicating exposures. The exposures are between 80 and 120 depending on the exposure. And then we, after 21 days, did a GGT, and after 30 days of exposure and extended light cycle exposure, did sacrifice the mice, took the metabolic active tissue, the adipose tissue, skeletal muscle, and liver, and measured systemic insulin resistance in these organs. So again, we repeated the experiment with normal mice, which were not exposed to anything else than PM, and there was again no change in glucose tolerance. However, when we exposed the mice on an extended light cycle, either exposure in the morning or in the afternoon induced glucose intolerance. Similarly, HOMA-IR was increased for both time points, and also HOMA-beta, indicating that there's insulin resistance and beta cell dysfunction. So now we wanted to know why. So how can this be? What's happening in the body? So under normal conditions, glucose homeostasis is regulated by the pancreas and the insulin release in the pancreas, which then binds to the insulin receptor, induces the phosphorylation of AKT, and then translocate GLUT4 and 2 to the membrane, which then facilitates the uptake of glucose in the skeletal muscle and also regulates hepatic glucose production. However, when we have insulin resistance, this pathway is impaired, leading to a decrease in glucose uptake and increase in glucose in the circulation. So we tested in all the 3 organs, and while we didn't find anything in the adipose tissue, we saw an impaired insulin signaling, further impaired insulin signaling in the skeletal muscle, as well as in the liver of mice that were exposed to an extended light cycle, as well as PM 2.5, indicating that impaired glucose uptake by the skeletal muscle, as well as the dysregulation in the hepatic glucose synthesis in the liver, are responsible for the decrease in glucose tolerance. So our previous studies have shown that pulmonary oxidative stress is something which is responsible for most of the effects we observed after PM 2.5 exposure. So we tested this here too. We are using ECSOD transgenic mice, which lung-specifically overexpress the human ECSOD, shown here. So there's no overexpression in any other organs, as we tested here in the aorta. We also found that these mice were protected against pulmonary oxidative stress, shown here below, by measuring GSH levels. So while in CAP-exposed mice, GSH decreased, we could restore GSH levels in these mice. And also the T-pores was indicating lipid peroxidation was, the increase was prevented in these ECSOD transgenic mice, indicating that these mice are protected against PM 2.5 induced pulmonary oxidative stress. So then we performed a glucose tolerance test in these mice, which this I only show the mice which were also exposed to the extended light cycle. And in the wild type mice, we see again that there was an increase in glucose intolerance. which was prevented in the ACSOD transgenic mice, indicating that this is regulated by oxidative stress. Similarly, the insulin resistance in the skeletal muscle and as well as the liver was prevented in these mice, again supporting our idea that everything is stimulated by pulmonary oxidative stress. We don't know exactly how, but we did some plasma experiments where there seemed to be a plasma factor which transfers something from what happens in the lung into the organs. We still have not figured it out 100%. It's like looking for a needle in the haystack. So we did also thought, okay, we should look in female mice. But surprisingly, they seem to be protected. So this is the same protocol. We had mice which were on a regular light cycle, female mice, and female mice which were on an extended light cycle, and then we exposed these to PM2.5. And while we can see that there is like still the light-induced increase in glucose intolerance, PM2.5 exposure did not exacerbate the effect of the extended light cycle. We thought that was really surprising. However, and looked also in the tissues, similarly, they were protected against insulin resistance in the skeletal muscle and liver. So we thought, maybe it's because they are protected against the pulmonary oxidative stress. So we used the lungs of female and male mice and looked for GSH and T-bars again. And while we see an increase in T-bars in the male mice, there was nothing to see in the female mice. Similar, GSH got depleted in the only in the male mice, but there was no change in the female mice. And this leads to an overall decrease in the GSH, GSSG levels in the male mice, but not in the female mice. In contrast to the depletion of GSH, we see in GSSG increase. So this could be maybe a dysregulation or an upregulation of pulmonary GSH, which protects these mice. We also looked for by PCR for other for the expression of other anti-inflammatory enzymes, and we found that SOD1, SOD2, and SOD1 to 3, as well as GST alpha and P are upregulated, and in addition to the regulator of antioxidant genes, gene expression was higher. So this indicates that female mice probably protected by against the metabolic effects of PM2.5 by preventing pulmonary oxidative stress induced by PM2.5. So we now found that that extended light cycle exacerbates the effects of PM2.5, but the question is still how. So we know the muscle and the liver get affected, and that pulmonary stress is involved, but how does it makes it to, what's the mediator of this whole thing? So our previous study showed that PM2.5 by itself induces endothelial insulin resistance and inflammation, which is known to be involved in metabolic disease. And we also looked in the extended light cycle and found a similar result. So extending the light cycle seemed to induce vascular insulin resistance. And it is known that the endothelium is a sensitive target for PM2.5 exposure. In addition, it has been shown that in insulin resistance, vascular insulin, in this case, diet-induced insulin resistance, vascular insulin resistance occurs far before skeletal muscle, liver, or even the adipose tissue get insulin resistant. So in addition, it has also been shown that endothelial insulin signaling is sufficient to impair glucose uptake in the skeletal muscle. Hence, we hypothesize that the endothelium and endothelial insulin signaling could contribute to the enhanced glucose intolerance in PM2.5. An overview how we think it should work is, so PM2.5 is inducing oxidative stress in the lung. This then activates NF-kappa and insulin resistance in the vasculature, decreasing the phosphorylation of AKT and the production of NO. This leads to vascular dysfunction, although it's no frank dysfunction, but this could impair perfusion of the skeletal muscle and lead to a decrease in insulin delivery, thereby, in addition with circadian desynchrony, impact glucose tolerance. However, still, how does PM2.5 in the lung induce vascular insulin resistance? Interestingly, vascular inflammation and insulin resistance have been associated with the disruption of the circadian clock. And as I showed you in the initial slide, so we have the central clock, which can impact the circadian rhythm and disrupt it. But there was also the possibility that a disorganization by, for instance, eating at the wrong time can impact the clock. And it has also been shown that several chemicals impair or interfere with the circadian rhythms. So this is important because circadian clocks are not only important for the peripheral clocks are not only regulating their own transcription, but they're also important for regulating antioxidative defense as well as inflammation. So the disruption of the vascular circadian clock is involved in vascular inflammation, impaired vascular signaling, a decreased insulin-mediated endothelial nitric oxide release, and it has been shown to alter glucose homeostasis. So we thought vascular inflammation and insulin resistance could be induced by disrupting of the vascular circadian clock. And to test for this, we exposed mice against to PM2.5, this time without any additional impact, because we wanted to see just the effect of air pollution on the vascular circadian rhythm. And we analyzed these the tissues we collected by the Cosinus method. So we took tissues around the clock every 4 hours for 6 times. And then perform PCR, which this was analyzed by the Cosinus method to determine whether they are following a circadian rhythm. And interestingly, we found that circadian genes were impacted by PM2.5 exposure, as well as oxidative genes, antioxidant genes, enzyme genes, and inflammatory genes shown here. So all over, I hope I can convince you that PM2.5 exposure in addition with this leads to disruption of a vascular clock, which then can reduce vascular inflammation and insulin resistance, inducing vascular injury, and in combination with either diet-induced obesity or circadian desynchrony, induced glucose intolerance. So we have 2 coins of a medal, right? We have on one point disturbed circadian rhythm as an environmental factor to increase the risk for cardiometabolic disease, and it increases susceptibility for air pollution exposure. And on the other hand, air pollution could be a circadian rhythm disruptor. As this is a CIEHS seminar, I wanted to highlight in the end collaborations with different labs. So I have a long-standing collaboration with the Conklin lab. We have published papers since 2012 on PM. I think this was the first one. Then I had worked with Dr. Sansbury together on air pollution and spleen issues. I had collaborations with Dr. Hill, which led also to an R1. And then with the O2 lab, we have recently published a paper on aging, and with Daniel Gomez as a first author. But also the CAR lab I got also involved in work with the CAR lab, doing some work in the heart. Dr. Merchant got some kidneys for his kidney project from our exposures. And then finally, we have collaborated with the Hellman lab, which leads to a CIEHS pilot program, pilot award. And in the end, I want to thank all the people who are either in my lab or collaborated with me. The female study was mainly performed by Michael Kitchen and Ashley Wright, who was a summer student last year in my lab. And then Amanda Ribble, who is doing almost everything from Western blots to carrying mice around, and all the other people who supported my career so far. Thank you so much, and thank you for your attention.
I'm sorry, did you look at over-optimized female mice?
No, we didn't.
Thank you.
I'm not sure how much estrogens plays a role, but this is definitively something we want to look in the future.
And I guess in a similar vein, your females showed some protection, and I assume they were the same age as the male mice.
Yes.
If you used older mice, older females that might mimic a postmenopausal state, or are they also protected?
So the problem with mice is they don't get postmenopausal. So it's a human thing. But we could use older mice. We have done older mice, but only males so far, because we started off with the male because we didn't saw any effects in the females. But we are planning to do female studies with older mice. That's in principle a similar approach than the ovarectomy.
Thank you so much. Great talk. So a lot of your research mentioned obesity and type 2 diabetes. Are you aware of any studies that have shown exacerbation of glucose tolerance or beta cell dysfunction in a type 1 model by chance?
I know there are some papers out which show that air pollution exposure increases the risk for type 1 diabetes. I cannot tell you exactly the mechanism right now on top of my head, but there were some associations found with type 1 too. Although autoimmune type 1 diabetes is an autoimmune disease, right? So there is a relationship between PM 2.5 and autoimmune disease as well as with circadian rhythm.
I have a couple of questions, and for the first one, and you're look like you're a PM, and you never mentioned what's the content. Have you measured the content? Is there any metal involved?
There were metals, yes.
You did that.
PM 2.5 is in principle a carbon core with metals attached to it, right? And pHs as well. So it could be that there is. I'm even almost sure because the metals would be the ones who maybe induce the oxidative stress in the lung. The Fenton reaction comes
So,
to mind.
but which metal is higher? Did you pay attention? Which one is cadmium or?
I know that there is a high content of iron in it, but there is also.
It's okay, and we can
It's
discuss
really
later
hard
for
to
that.
tell.
Yeah.
It's a big mixture of metals.
Yeah, we can discuss that later. Second question is that you are looking for the insulin signaling pathway, and you only mentioned the total AKT. And I know that like insulin, like a glucose metabolism is a predominant rely on the type AKT2. Have you looked for the different isoformed
No.
AKT1, AKT2?
We use a pan antibody. I have not figured this out in this detail.
That's fine. Yeah. And the third question, you have one transgenic mice you're using pulmonary and overexpressed SOD. Is that endothelium or a fibroblast or a epithelium cell? Which kind of cells?
It's type 2 epithelial cells. It's expressed, right? It's a type 2 epithelial cells in the lung which overexpress it, and no other cell type other than this. So it's a promoter specific to
specific for the lung. Okay, that's excellent. And the last question, and is you are like a very very surprised, unexpected, very surprised found that the female is totally different from male. But the two different experiments is not the same time. Your exposure probably different than PM. And you know the collected PM, different time have different component. How do you explain to that?
So yeah, it could be, but normally the composition doesn't vary too much when we do this here in Louisville, except when we have maybe yesterday. I'm pretty sure yesterday should be looked at a separate experiment at all, but also with the wildfires when they come in, then it can maybe change a bit, especially in the concentration. But usually especially when these were done, there were not much variations, and most of them are done in the summer. We compared one time, and there was not a big deal with the composition. With the females, they were exposed to 60 microgram per meter cube that usually should be sufficient to induce
So that is like
glucose intolerance.
kind of explain the question, but I think if in the future, if I have like a similar experiments, and if I have additional tissue, we are willing to help you to detect the metal in the tissue to determine if that is a kind of exposure to metal have caused the difference or not.
When you can use frozen tissues, we have
You
a file.
have? Okay, yeah, we can think about that. Yeah.
Yes. Yeah.
Thank you.
Yeah.
I'm sorry, have you looked at any humans that are working night shift, such as at the hospitals and stuff? Because they're essentially reverse light adapted. They're sleeping during the day and
I mean, there is people who are doing night shifts are more prone to type 2 diabetes. Yes. And all of this is, they also tested it and adjusted it for eating at the wrong time, right? When you do night shifts, you also eat at the wrong time, but there was an association just with the shift and not with the eating habits.
Kind of related question, more kind of basic fundamental question, because mice are nocturnal. So extended light exposure, they are already adapted to sleep under light. And I wonder how the extended light exposure to mice, so I mean, how the findings can be translated into humans.
It seems very counterintuitive. And we did this initially when we started the whole experiment because it was easier to accomplish. However, there's a reason too. When you remember my slide, it does not matter whether you are awake longer or you sleep longer or shorter. Both of it would affect your risk for type 2 diabetes. So we thought it is an appropriate thing to do this. Yes, I agree. It feels counterintuitive with the nocturnal mice. Yes.
Thank you.
Yeah, thanks, Petra. And you left us plenty of time for questions, which is wonderful.
You know, I speed up all the time.
Yeah. So
much slower yesterday.
I thought maybe we would address some of these questions that came up about composition that Lu Kai raised. Now, we've done a number of studies with just volatile organic compounds, no metals involved. And maybe Dr. Haberzettl could tell you about some of the results that we see in those exposures.
So we haven't done the big glucose tolerance test, but we have looked for vascular inflammation and insulin resistance after aqualine exposure. And aqualine inhalation also induces this. Then we looked for VEGF resistance and EPC's mobilization, and it looks like it's also impaired by both, by PM2.5 exposure and by aqualine. So there seem to be, I think there is a pulmonary oxidative stress underlying mechanism maybe who triggers volatile as well as, but I cannot really point this 100% to this. But still, I think the oxidative stress in the lung could be triggered by metals in PM2.5.
No, and that's a great lead in to the pulmonary protection hypothesis, because you saw changes in antioxidant genes. So one of the questions is, what is the circadian rhythm of antioxidant responses just normally in people or in animals?
So it follows, so normally you're in humans as you are regulating up your antioxidant defense enzymes during the day and lipid oxidation products resolving things in the night, so that you kind of battle the oxidative stress during the day as a human and try to deal with the products in the night. When you look here, so this is the full line is actually the expression of, in this case, SOD3, and then this would be nerve 2. It follows a circadian rhythm. So it seems that the circadian clock regulates the expression of antioxidant enzymes. Does it? In mice, it's
So-
done the opposite, right? So mice have in the night upregulation of antioxidant enzymes, and
So you're showing the mRNA here.
yes, I know.
So the reality is you want to see the protein, right?
So we looked at proteins too. Problem is, this is vasculature. So we did this experiment not too often because it's a 24-hour experiment, and I have sometimes hard time to convince people to stay for 24 hours in the lab. So we collected the aortas and did PCR. We did with aortas which were not collected at all the time points, Western blots, and we saw changes in SOD2, SOD3 too. So there is some protein changing as well.
Well, I agree. The vasculature is more difficult to look at the protein levels, but what about the lung?
We looked in the lung too, and it was increased.
Well,
The
what was,
protein, we looked for SOD for all the 3 SODs, and SOD3 was increased as well.
so I'll just share an anecdote. In our tobacco studies, we used male and female mice, and we put them on a shifted light cycle. So that is, we were able to have mice in the dark during the day, which would be their normal active phase. And then we had the other ones just on the regular light cycle. And we exposed them for 12 weeks to cigarette smoke, either during the day for the one group or in the dark for the other. And the ones in the dark were quite active. Like, we could see them running around. And for anybody who's done exposure studies during the day, the mice just more or less go to sleep. So our hypothesis was that the increased activity during the dark cycle was going to increase their uptake of cigarette smoke. And in fact, what we saw was exactly the opposite expected result. That is, the mice exposed during the dark had less atherosclerosis than the ones exposed during the day when they were sleeping. And in fact, what that really fits really well is that if the antioxidant responses were basically shifted with that time change, it could give one explanation why the mice were protected during the dark when they were more active, which I think is really an important situation that we're addressing here. If you're supposed to be sleeping and your antioxidants go down and you're up working and being exposed to light and all these other things, it creates a mismatch.
We did a similar experiment with the PM 2.5 exposure as well. We looked for vascular inflammation. Interestingly, it was lower for the mice which were exposed during the night. But I found that glucose levels, for instance, were higher in the mice which were exposed at the night compared to. So there seems to be some protection when you expose mice in the dark, but not fully. So I'm working in the moment on getting all the data together for a manuscript. So there seemed to be an issue, and there seem to be different effects whether you're exposed at day or night. Yes, and it could be protection for this one and not for this one.
No, I really like the idea of linking it with glutathione, because the GSTs, and especially GSTP, protects mice against cigarette smoke and acrolein-induced endothelial dysfunction.
Yes, you remember we had really controversial things with the GSTP mice at one point, and the TRIP1. I remember, yeah, we did this.
Yeah. No, GSTP definitely protects against no matter what time of the day. So in the knockouts, it would be interesting to show how much more sensitive they are, and they don't actually upregulate it at any point. So.
Thank you.
Okay, if there's no one else, I've got one quick question and then we'll check online. So you noted that really interesting sex difference where the females seem to be protected, but If we talk about cardiovascular disease, obviously males are affected with greater incidence than women. And if we look at epidemiological data, there's links between disrupted circadian rhythm and increased incidence of diseases, more so linked to women, like Alzheimer's disease and breast cancer.
Mhm.
So kind of at a broad level, is there any difference in the vulnerability of these peripheral clock genes across tissues? So is it different in the brain compared to